Hi guys, so today we'll be doing an experiment entitled Banana Stripper. And no, we won't just be peeling off this banana. But we will extract the DNA out of it. So before the experiment, what is DNA? DNA, or deoxyribonucleic acid, is a material that holds the blueprint or information that tells your body how you will grow, develop, and behave in a certain way. These characteristics are expressed by living things, such as bacteria, banana, and human, and all other living things. In short, all living things have DNA. Also, information are passed from one generation to the next. Notice that each organism has its own character, its traits different from the other, or how this banana is different from the other types. Simply, how you got your long fingers from your father or your hazel eyes from your mother. This means that you've got the same part of DNA from them. So where do we find it? It is present inside the nucleus of all cells except matured blood cells. And DNA gets replicated for the daughter cells when a cell undergoes cell division and get encoded into protein with a function determined by its sequence. So what does it look like? Under a high-powered microscope, you will see a double helix-shaped molecule that resembles a spiral ladder. It is also composed of long nucleotide strands. So their structure can only be seen under the microscope, but we can actually extract them as a whole to make them tangible. Without further ado, guys, tara na! For this experiment, you will need food processor, salt, dishwashing liquid soap, a narrow glass jar, a knife, a strainer, a banana, and a toothpick. After gathering your materials, using your knife, cut half of the banana into tiny pieces to expose more of the cells. Then place your banana pieces in the blender, add a teaspoon of salt, and slightly cover the mixture with warm water. Mix in the blender for 5 to 10 seconds, making sure the mixture is not too runny. Next, pour the mixture into the glass jar through the strainer. And add about 2 teaspoons of liquid soap. gently stir the mixture. Try not to create bubbles when stirring. Carefully pour very cold rubbing alcohol down the side of the glass topping near the top. Make sure that two separate layers are being formed. Wait for 5 minutes to allow the DNA to separate from the solution. Lastly, use the toothpicks to extract the DNA that floats to the surface. Now that we've finished our experiment, let's go through the 4 general processes that we've just performed. The first process. The mashing process increased the surface area or simply exposed more of the cells. The purification process using the strainer helped us filter out the impurities such as the large particles of the slush banana that may have hindered us in extracting the DNA. The lysis process occurred when we poured in and stirred the liquid soap. This helped us in breaking the cell membranes, exposing the inner structures, including the nucleus. Now, DNA is naturally hydrophilic, able to keep the DNA intact using the salt. How so? The negative charge of the DNA interacted with the positively charged atom of the salt, which is the sodium, instead of interacting with the positively charged atom of the water molecule. The precipitation process explains the aggregation of DNA molecules after pouring in the cold alcohol. Following that DNA is insoluble to the alcohol, it is termed as precipitate.